क्या हो रहा है यार? हम्म, हम्म, Something is the issue. So good evening, good afternoon. Um, Today we will be looking at the content of week 9 and then look at few questions. So to begin with, in order to characterize a product based on the molecular weight, we can have gel filtration chromatography, which we looked at earlier in the earlier tutorial session, or SEC. We can use electrophoresis, whether be it using different kinds of matrix like acrylamide, polyacrylamide gels, agarose gels, or a mixture of agarose, polyacrylamide gels, etc. One can even do mass spectrometry to look at the composition of the suppose for protein mass spectrometry, one can even look at the sequence of amino acids in a peptide. So from gel filtration chromatography, one can have an idea about the native molecular weight of the like um, of the protein in question, or the biomolecular macromolecule of interest. From electrophoresis, one can even have the native um, molecular weight and also the unit subunit molecular weight as well. Okay, like suppose in a native conditions, something exists as a dimer. However, upon electrophoresis like SDS page, which is kind of a denature, which denatures the dimer, and proteins exist as monomer in like in an SDS sample. So we can eventually get the uh, molecular weight of the subunit in there. From mass spectrometry as well, one can get both the native and subunit molecular weights. To identify the type of functional group that is there, one can have IR and UV visible spectroscopy. These methods can be used, and one can even sequence the suppose what is the sequence of a nucleic acid stretch, what is the genetic sequence. One can even have the RNA sequence or the DNA sequence as well. Okay, now this we looked at in the earlier tutorial class as well. Let's just brush it up once more. How can we really use gel filtration chromatography or SEC for determining the molecular weight? Now, SEC involves SEC actually partitions the biomolecules in a sample according to its molecular weight. The type of molecule that has the highest molecular weight can't really enter through the pores of the matrix, and it passes out and mostly comes out in the void volume, and the ones which are really smaller do have access to the pores, and hence it takes a longer time to elute out of the SEC matrix. All right. So, so accordingly, if the one that elutes out first is the largest molecular weight sample, whereas the one that elutes out in the end has the least molecular weight out of the like in the complete mixture. So this is what, so if we see from this profile, 6 has the least molecular weight, so it comes at the end, elutes out at the end, and sample 1 or molecule 1 has the highest molecular weight. Okay, so this is 1 2 3 4 5 6 is the decreasing order of molecular weight of the sample or molecules in the mixture. And the molecular weight and size of a protein is related to the shape of the molecule, and it can be related, like it is directly proportional to like the molecular weight, the radius of gyration is directly proportional to the molecular weight and raised to the power of a particular constant, and this particular constant actually varies depending on what is the shape of the molecule. Okay, so it can have different values for a rod shaped molecule, for spherical molecules, etc. One can even have the oligomeric status of the protein. Okay, so to have the oligomeric status of the protein, we have the same SEC profile, suppose, and it is eluting out. Suppose the protein um, elutes out, our protein of interest always elutes out at this elution volume, right? So we always get our protein in this peak. Although if according to the, and so corresponding to this particular elution volume, the molecular weight of the protein is suppose 30 kDa. However, when we run the same sample in an SDS page is a denaturing page, right? So the protein, our protein of interest, runs at 10 kDa molecular weight. Okay, runs corresponding to the band of our protein of interest comes near the band corresponding to 10 kDa protein. Okay, so that means it is forming a trimer, right? So how do you really know how many, like what is the oligomeric status of the protein? So first, this is in the native condition. Okay, molecular weight gel filtration, whatever we are getting, the corresponding molecular weight corresponding to this elution volume divided by the molecular weight after running SDS page. Okay, so this gives us the oligomeric status of the protein. Next, we can also study protein folding, where in the presence of a denaturing agent like urea, if we keep on increasing the concentration of urea, what will happen is a well folded, properly folded protein structure will eventually slowly start unfolding. Okay, so as if a protein structure is properly folded, it will, the overall hydrodynamic radius or hydrodynamic surface of the completely folded native protein structure will be comparatively less as compared to the hydrodynamic radius or hydrodynamic surface of a partially unfolded or a completely unfolded molecule, right? So similar to like how supercoiled plasmid DNA like is the smallest, right? The hydrodynamic radius of a supercoiled plasmid DNA is the smallest because it's coiled upon itself, like, and then it can easily move through the pores, right? So if a native protein which is properly folded, overall surface area is less, hydrodynamic surface area is less, hydrodynamic surface is less, and hence it can get access into the pores of the matrix and takes a long time to elute out of the SSC matrix. However, if as compared to the fourth one where it's completely unfolded, the overall radius increases, hydrodynamic radius, and hence it has limited access to the matrix pores, or in some cases no access to the matrix pores, and hence it comes out earlier, elutes out earlier out of the column as compared to the one that is in the native condition. Okay, so in this way one can even check the oligomeric status of the native protein in the presence of a denaturant like urea in this example. One can even see like check protein ligand binding. So if a ligand is binding to a protein, overall size or overall hydrodynamic radius of the complex will be greater as compared to a single ligand or single protein, right? So what will happen is the peaks, suppose usually if it's a small ligand bound to a protein, like comparatively larger protein, so the ligand will elute out at the end, followed by the protein, followed by, if there is some interaction between the ligand and the protein, then that ligand protein complex will elute out at the very beginning. Okay, uh, because it's comparatively larger and it won't have access, or it will have limited access to the pores of the SEC matrix. Okay, this is what, as the concentration of the ligand is increased, protein binds the ligand and forms a larger complex with an increase in the hydrodynamic surface area. As a result, protein peak shifts towards the left. Okay, now in general we have been talking about agarose, electrophoresis and SDS page. So basically, electrophoresis is a general term that depicts the migration and separation of charged particles affected by an electric field. An electrophoretic framework comprises of two terminals, one cathode and one anode, okay, having opposite charges associated by a directing medium called an electrolyte. Right, there should be some movement of ions which will eventually help in the like passage of electricity, right? So the separation impact on the ionic particles results from contrasts in their speed, one, which is the, and why, where does this come from? This depends on the portability M and the field strength E. How strong is the applied electric field or electric strength? How strong it is? Strength of the electric field, like basically measured by the voltage that we apply, okay, while running the gel, whether be it SDS page or agros, and what is the overall size of the molecule and overall molecular weight of the biomolecule in question. Okay, so if this is uh, the electric field, and this is the, so also while moving in, like during electrophoresis, when your sample is moving, there is the electrostatic force, definitely there is electric uh, the strength due to electric field as well. Also there's a viscous drag on the opposite side faced by the molecule in question, the sample that we have loaded on the gel. So the portability M of an ion is not entirely set in stone by molecular size, shape and charge and the temperature during the separation, and it is consistent under the characterized electrophoretic conditions. Okay. So this is what is again the same depiction. If this is your this is the molecule that is moving, so this particular uh, molecule is experiencing two types of force predominantly. One is the force due to the electric field, okay, which is charge multiplied by the electric field strength, and there's a viscous drag on the opposite side. So these two forces basically um, influence the electrophoretic mobility. So electrophoretic mobility is directly proportional to the charge and inversely proportional to the viscosity of the medium. Okay, and the size and shape of the molecule denoted by R. In this case of relative mobility, it is directly related to the charge by radius E by R of the molecule. Now if we talk about a globular protein, suppose the radius of the molecule is related to the molecular mass of the macromolecule, which is molecular mass of the macromolecule. Okay, now there's something called as moving boundary electrophoresis. Okay, this is carried out um, like eventually when we came to today's very commonly used electrophoretic methods. So there were different kinds of other electrophoretic methods. One was moving boundary electrophoresis, which is carried out in a U-shaped tube with platinum electrodes, right, attached to the end of both arms, these two here. And at the respective ends, a refractometer is attached, which basically monitors the changes in the refractive indices. Okay. Um, and sample is dissolved in the buffer and loaded in the middle of the tube here, and then the apparatus is connected to a power supply. Now what happens is charged molecules will move to the respective oppositely charged electrode, right? Positively charged electrodes will move here and negatively charged electrodes will come here. Okay, and the sample is dissolved in the buffer, and charged molecules move to the respective counter electrode and passes through the refractometer. Now the refractometer actually measures the changes in the refractive index of the buffer due to the presence of the molecule. Now when the molecule of our interest is passed, sample is taken out from the apparatus along with the buffer. So this actually is used, or more, it was used to study the behavior of the molecule in electric field earlier, and definitely now this is not used very much. Okay, camera is shut. but the resolution of this technique is low due to mixing of the sample as well as overlapping of the sample components. And there is something called as zone electrophoresis, where a strip is used to connect two different um, electrodes. Basically, it involves the migration of the charged particle in question on the stabilizing media, and components are separated, which are distributed into discrete zones on the supporting media here. And the supporting media is actually saturated with buffer solution, and small volume of sample is applied as a narrow band. And on application of the sample at the ends of the strip here, this one, components migrate at the rate determined by the electrophoretic mobility, and the separated colored components can be separated by electropherogram. Okay. Now this is something we discussed in a lot of detail in few tutorial sessions back. Again, we can briefly go through it, which is an overview of SDS page electrophoresis. It has two types of gels. One is the stacking gel and the resolving gel. And the main thing that needs to be taken care of is the pH here, which actually plays a huge role in separating the proteins out, okay, on the gel, acrylamide, bisacrylamide matrix. pH 6.8 stacking gel helps in stacking all the protein molecules at one point, and resolving gel 8.8 finally helps in separating the sizes, like different sizes of molecules. Okay, and the mixture of macromolecules are applied. The one here, the pink one here, is the smallest one, which will eventually move the fastest, and the largest one will move the slowest, okay, on an SDS page. This is a polyacrylamide gel matrix. So how it works, it separates the proteins according to their molecular weight based on their differential rates of migration through a sieving gel under the influence of an applied electric field. And movement of any charged species through this field is actually determined by the net charge, molecular radius, and magnitude of the applied field. So all this complete set of slides are also there in the previous tutorial sessions. And in SDS page, what is the why do we really need to do SDS page? What is the problem with natively folded proteins? Because their net charge nor the molecular radius is dependent on the molecular weight, and net charge of a protein in question depends on its amino acid composition. The side chains of amino acids can vary, and based on the pH of the surrounding medium, the overall charge on one amino acid can differ. So if each amino acid shows different behavior in different pH, then overall charge on the protein isn't really uniform. So the sum of positively and negatively charged amino acids in the protein and molecular radius by the protein's structure vary by the actually um, this is the net charge that actually influences these influenced by positively and negatively charged amino acid residues. So in a native state, what happens? Different proteins with the same molecular weight can migrate at different speeds in an electric field depending on their charge and 3D shape. Now, to separate these proteins in an electric field based on their molecular weight only, one needs to destroy the tertiary structure, the overall properly folded structure, by reducing the protein to a linear molecule, and also mask the intrinsic net charge of the protein. So the different buffers and reagents for electrophoresis are, one is definitely you need the buffering agent, which is tris-Cl or tris-HCl. You need glycine. The different charged states of glycine is actually used um, to form the different running fronts. Like one is chloride ions, one is the different charged states. Like glycine has a different charge. Like overall, I'm talking about the net charge on the glycine amino acid. It has a different charge at 6.8 pH and different charge at 8.8 pH. All right. So glycine is actually, so if we talk about the gel in question, so the gel in question actually has tris-Cl, stacking gel of different of pH 6.8, resolving gel pH 8.8. Okay, then you have to pour acrylamide bisacrylamide solution at a particular ratio, like how much ever pore size you really want. and then one puts SDS goes in there, and there is something called ammonium persulfate, which actually provides the free radicals for polymerizing the acrylamide. Okay, and it isn't only acrylamide, bisacrylamide is the cross-linker here. Okay, acrylamide is the monomeric unit, and each monomer is actually cross-linked or branched. This branching occurs due to the presence of bisacrylamide here. Okay, and when this is the overall, and then timid is something which acts as a catalyst, and it catalyzes the polymerization of the acrylamide. Based on how much timid you are adding, it actually either fastens the process, uh, fastens the solidification process, like mainly acrylamide polymerization process, or if you add it in a proper, at the properly specified volume or amount, it actually polymerizes nicely in a proper, the rate of polymerization is proper, and the regulation of the pore sizes is also proper. Okay, and then comes the running buffer. Okay, so where does glycine come from? Glycine comes from the running buffer. Running buffer is TGS, which is tris. Cl is there in the running buffer, tris is there, then glycine is there, and S stands for SDS. Okay, and then these ones, bromophenol blue, Kumasi brilliant blue, these are used in the detection of the molecule. One is bromophenol blue, which is, there is something when we prepare the samples, we add a tracking dye as well to monitor how far on the gel. The gel is a transparent matrix. Okay, so on the gel, to visualize how far the samples have run, bromophenol blue is the tracking dye. Okay, and then there is something called Kumasi brilliant blue, which is used to stain the polyacrylamide gel, and Kumasi binds to the protein of our interest and stains it blue. Okay, so ammonium persulfate in the presence of timid actually forms oxygen free radicals and induces the polymerization of acrylide monomer to form a linear polymer. All right, and these linear monomers are interconnected by cross-linking with bisacrylamide monomer to form a 3D mesh-like structure with pores. The size of the pore is actually controlled by the concentration of acrylamide and the amount of bisacrylamide in the gel. Now, in a vertical gel electrophoresis system, which we have been looking at this constant way, agarose gel electrophoresis mostly was like this. The gel is running this way. But vertical electrophoresis system, mostly SDS page, runs in this manner. Okay, as in vertical manner. I don't know if I'm visible or not. Um, but yeah, first the resolving gel solution is prepared and poured into the gel cassette, because that is what lies below. And then on top of the resolving gel, stacking gel is poured. Right. So the first one to actually, for the samples, the first portion is the stacking gel that the sample molecules encounter. However, while preparing the gel, we first have to prepare the resolving gel and pour it, followed by, once the resolving gel solidifies, then one pours stacking gel on top of it. Okay, many times for after pouring the resolving gel, a thin layer of organic solvent, any kind of alcoholic solvent, mostly isopropanol, is layered on top to prevent the entry of oxygen. Because what happens is ammonium persulfate is like forming oxygen free radicals, right? So if there's the oxygen present in air, neutralizes the free radical and slows down the polymerization. So that's why we block the access of the gel to the air, to the oxygen present in the air, so that are also adding isopropanol. Actually, if we introduce any bubbles in between, it also it helps to remove the bubbles from there. So after polymerization of the resolving gel, the stacking gel is poured, and the comb, which eventually forms the wells, are fitted into the gel for construction of different lanes for loading the samples. So SDS basically is the detergent present in the sample buffer. We boil the sample, and also the sample loading dye has a reducing agent like DTT or BME to break down the disulfide bonds. Eventually, all this treatment disrupts the tertiary structure of proteins, and folded proteins are uh, broken down to linear molecules. So SDS actually coats the protein with a uniform negative charge, which masks the intrinsic charges on the R group of the amino acids, and binds SDS actually binds quite uniformly to linear proteins around 1.4 SDS for a gram of protein, which means the charge of the protein is now approximately equal to, like, proportional to its molecular weight. So now the dominant factor in determining an SDS coated protein's mobility is the molecular radius, basically is the size. So SDS coated proteins have been shown to be linear molecules, approximately 18 angstroms wide. This need not necessarily be applicable for all types of SDS coated proteins, but generally it has been seen that it is 18 angstroms wide with length proportional to the molecular weight. Okay. So this is the stacking gel, which is low concentration of acrylamide. Proteins do not separate here, and then the running gel has high acrylamide and capable of retarding protein movement. And stacking gel has a pH 8. Glycine, which we talked about, which comes from the running buffer, it switches predominantly to the neutrally charged state at this pH, and hence there is a loss of charge causing the glycine ions to move very slowly in electric field, and chloride ions from tris Cl move much more quickly in electric field and form an ion front migrating ahead of glycine. And separation of chloride from tris counter ion uh, creates a narrow zone with steep voltage gradient. So all the proteins in the gel sample have an electrophoretic mobility lying in between the extreme mobility of glycine and chloride. And two fronts sweep through the sample well. These are the sample wells here. And proteins are concentrated in the narrow zone between chloride and glycine fronts. So this goes on until the resolving or running gel uh, has, like, the sample molecules hit the running gel, which has a pH of 8.8. Now glycine molecules become mostly negatively charged and it migrates much faster than the proteins. So glycine front accelerates past the protein. Proteins are now dumped in a narrow band at the interface of the stacking and running gels. And for running gel, increased acrylamide concentration also slows down the protein movement, and hence the resolution or separation begins according to the sizes of the molecules. So why are we basically using the stacking gel? Gel wells are nearly 1 centimeter deep. It needs to substantially fill uh, to have enough protein on the gel. And if we are in pouring stacking gel, then the sample would actually sit on top of the running gel as a band of up to 1 centimeter deep. And rather than being lined up together and hitting the running gel together, proteins would all enter the running gel at different times, resulting in very smeared bands. Okay. So stacking gel ensures that all of the proteins arrive at the running gel at the same time. So proteins of the same molecular weight will migrate as tight bands. So based on the percentages of acrylamide, if the percentage is lower, that means the pore sizes of the polyacrylamide gel are bigger. Okay, so we can run bigger molecules with higher molecular weights. Whereas if the percentage is higher, so that means the pore sizes of the polyacrylamide gel is more regulated and more tightly regulated, and the molecular weight can range from 3 to 100 kDa. So loading die I was talking about is a sample buffer used to prepare and load protein samples on the SDS page gels. It contains a number of components including SDS, which denatures the proteins and adds a negative charge to them, allowing them to move them in an electric field. There's bromophenol blue, which is a tracking dye that allows to visualize the proteins as they run on the gel. Not really, we are directly visualizing the proteins here. We just can monitor how far our sample has run. Bromophenol blue doesn't really stain the proteins. Okay. Then there's glycerol, which again increases the density of the sample, and hence so that the sample can settle down inside the wells in a better way. And tris helps to maintain the pH, and there's this reducing agent BME or DTT, which breaks the disulfide bonds and keeps the proteins unfolded. And then after the gel run is finished, Kumasi brilliant blue stain is used, which helps in routine visualization of proteins separated on polyacrylamide gels. This is an organic dye that makes complexes with basic amino acids like lysine, histidine, tyrosine, and arginine, and overall negative charge to the proteins, allowing their separation on the polyacrylamide gel. And different kinds of Kumasi dyes are actually available, like Kumasi Brilliant Blue R250 or G250. R actually stands for the more red and G stands for more green in the dye. So how does Kumasi Brilliant Blue stain works? So basically this stain, suppose if we talk about Kumasi Brilliant Blue G250, it has three forms, okay? Anionic blue form and neutral green form and cationic red form. All right. So if the environment is acidic, which means there are more H plus ions, okay, so then the red form or the red dye, which is cationic, is converted into its blue form or anionic after binding to the protein of interest. Okay. Um, and then if no protein is binding to the dye, then the solution remains brown. All right. So the CBB stain forms a strong non-covalent complex with the carboxyl group of the protein by van der Waals force and the amino group like NH2 group through electrostatic interactions. Now during the complex formation, the red form of the Kumasi dye first donates its free electron to the ionizable groups on the protein, causing a disruption of the protein's native state and exposing its hydrophobic pockets. These pockets non-covalently bind to the non-polar components of the dye via van der Waals forces, and the ionic interaction further strengthens the bond. So, this overall complex enables the reduction of proteins separated by the gel. So, if we are staining with Kumasi Brilliant Blue, this is how it looks. It's actually completely very dark, purplish, bluish colored stain. And then what happens? There's a de-staining solution. Mostly this staining solution has Kumasi Brilliant Blue, methanol, uh, acetic acid and water. And de-staining solution has the same composition just with the exception of this Kumasi Brilliant Blue dye. And so after, this is the dye that has bound to the proteins bands. Okay, that was run on SDS page. So how do you really analyze an SDS page image? So this is the marker, reference marker of where proteins of different sizes are there. And you basically, this is your sample. You actually compare the migration of your sample with respect to the different bands on the marker lane. Okay. And make a graph here, and then you finally have the RF value which is actually calculated by dividing the mean distance traveled by the protein divided by the distance traveled by the dye. Okay, so the dye front will be there, the bromophenol blue that we were talking about. So in comparison to that, so suppose there will be D5, D4, D3, everything will be there, D2, D1, and then you will get a correlation, like correlated graph here. And then finally, based on, like, there will be a distance traveled by your protein here, uh, this RF value will be there, and hence you can eventually extrapolate it to get the molecular weight. Now SDS page, we have SDS page. Now there's something called native page, where SDS page, like, there's no denaturing conditions, there's no reducing conditions. We check the uh, gel profile of the native state of the native state of the sample in question, or native state of the protein in question. So what happens is, in general, if we talk about SDS page, it is a detergent. There's beta mercaptoethanol, which actually gives equal charge to all proteins, breaks the disulfide linkage. 3D structure of the protein is destroyed, and only the migration is based on the subunit molecular weight in SDS page. However, in native page, sample is prepared in the loading dye. It does not contain detergent or any denaturing agent, and as a result, the sample runs on the basis of charge by mass, because this charge is actually given by the intrinsic charges of the like constituent amino acids or constituent um, nucleotides. Okay, and divided by mass. So in native page, usually the 3D conformation is, as well as the activity of the protein, remains unaffected. There is something called urea page as well. So any insoluble protein is dissolved in urea, and the samples separate based on their charge by subunit mass. And gradient urea page. We looked at gradient urea page in case of SEC as well, as we were increasing the concentration of urea, which is a denaturing agent. The native state of the protein was unfolding slowly. So gradient urea page is used to monitor the folding states of a protein. Now, how do you really prepare the sample? You add an appropriate amount of 2 times gel loading mixture to the sample, and the loading dye must usually contains formamide, okay, which again denatures any kind of structure present in your sequence. Then there's EDTA, which actually chelates the ions required for functional nucleases. And then there's xylene cyanol and bromophenol blue as tracking dyes, which runs at different speeds. And before the like bromophenol blue runs faster than the xylene cyanol dye front. And before the samples are loaded on the gel must be heat denatured. The samples must be heat denatured by heating between 70 to 90 degree Celsius for a few minutes. You guys can also check this reference, which talks about urea page. Okay. So, in general, while running different kinds of page, the band sharpness depends on several factors. It depends on how much volume of the sample you have loaded. If you're loading very small, like 1 to 5 microliter of sample, then you will eventually have really sharp bands. However, up to 15 microliters of sample can also be loaded. Um, but like in general, for a general case scenario, less sample volumes results in sharper bands. The band quality is also dependent on how much thick the gel is. Thinner gels, like as thin as 0.75 mm, show better results as compared to thicker 1.5 mm gels. Shape of the bands can also be influenced during gel loading. If the sample is not applied equally in the gel pocket, uh, what happens is, if glycerol quantity is not fine in the loading buffer or loading die, it might lead to unequal sample loading. So actually adding glycerol in the loading die helps during gel loading, and the sample settles into the well more easily. There can be another problem that is related with the sample loading buffer if the expected products run at the same level as one of the loading dies. So additionally, if fluorescent labels are used, some dyes show a fluorescent signal at a similar wavelength. If this is the case, removing one dye or all dyes can solve this problem. Then it is advised to run the dye containing sample in an empty well as a size standard. Also, low voltage at the beginning of the gel also helps to get sharper bands as the sample enters the gel front smoothly and it avoids the gel pockets collapse due to high voltage. A short, low percentage acrylamide stacking gel can have the same band sharpening effect. An often encountered problem is gel smiling, which is due to uneven heat distribution during gel electrophoresis. If the gel is not surrounded by buffer, depending on which gel system is employed, attaching of a metal plate to the glass plate supports an equal heat distribution and can substantially increase the gel quality. There is something called silanizing of glass plates for larger gels because it improves the handling of gels after the run as the gels tends to stick to the glass plates. You can eventually very easily take the gel plates out of the uh, glass plate, because otherwise if the gels are really long, then the gels get torn eventually while transferring it out of the glass plates and into the buffer or into the staining solution. Denaturing urea page is useful to analyze or separate single stranded DNA or RNA fragments as well as like um, ribonucleoprotein or nucleoprotein complexes. Now, there are protein ladders which are composed of a series of bands which are highly purified polypeptides of different well defined sizes. Now, these protein ladders get resolved during electrophoresis as separate bands based on their molecular weights in a pre-stained protein ladder, which are shown here. Okay, each of these bands or polypeptides are already stained with the dye. Even loading the ladders into the gel for electrophoresis, these ladders are previously stained and ready to go. What happens during electrophoresis? The polypeptides of the ladder show up in the gel as distinctly colored bands, like here. Okay. And even before the gel has completely run and is further stained with the gel stain Kumasi stain, the ladder bands can be visualized. This actually gives an idea of how far, suppose your protein is 20 kDa. So even if you have overrun your gel and lost these many bands, you can still be sure that okay, or suppose your protein is 75 kDa. So you are not really interested in any kind of molecular weight lower than 75 kDa. So you can actually overrun the gel and then just see how far this particular pink or red band has run, and then eventually take the gel out, like stop the electrophoretic run. So each band here, this lane molecular weight, like protein ladder, represents a molecular weight which is a combination of the molecular weights of the corresponding polypeptide and the associated dye, because all these have been pre-stained. Okay, so it actually has the associated dye weight as well. So this is the gel, pre-stained ladder in the gel, and then gel to membrane transfer. There can be two possibilities. Suppose many times one needs to probe the gel, probe your protein of interest with the uh, antibodies, okay, against the like antibodies can target your protein of interest that has run in STS page. So all the protein bands can be transferred on a suitable membrane. This is called Western blotting. Okay. So even with a pre-stained ladder, you can actually check how fine the transfer of your protein bands were. Suppose if it's very distinctly colored on the membrane, Western blot membrane, then you know that the transfer has taken place nicely. This is a good transfer. And if the bands aren't really visible properly, then it's a poor transfer. Many times there are unstained protein ladders as well, with bands or polypeptides which are not pre-stained, and hence it cannot be visualized while the gel is running. Visualization takes place after the entire gel is stained with the dye like Kumasi. Because no additional stain is added during electrophoresis, the representative weight of the polypeptide is more accurate. Hence, the size of each band in the ladder represents solely the molecular weight of the corresponding polypeptide constituent. Similarly, there's agarose gel electrophoresis, where agarose is a natural linear polymer extracted from seaweed that forms a gel matrix by hydrogen bonding when heated in a buffer and allowed to cool. And agarose is the most popular medium for separating moderate and large sized nucleic acids and has a wide range of separation. It basically separates DNA fragments by size in a solid support medium. Sample like DNA or RNA is pipetted into the sample wells, followed by the application of electric current, and rate of migration is proportional to the size. Smaller fragments move more quickly and wind up at the bottom of the gel. And DNA is visualized by including in the gel an intercollecting dye, which is ethidium bromide. DNA fragments take up the dye as they migrate through the gel. This dye actually stacks in between the bases of the nucleic acid, and illuminating with UV light actually causes this dye ethidium bromide to fluoresce. The larger fragments fluoresce more intensely. Although each of the fragments of a single class of molecule is present in equimolar proportions, the smaller fragments include less mass of DNA, take up less dye, and therefore fluoresce less intensely. A ladder set, same thing as marker that we looked at earlier, is a set of DNA fragments of known size which can be run simultaneously and used to estimate the sizes of the other unknown fragments. Loading dye again is a colored dye mixed with DNA samples before agarose gel electrophoresis to help visualize and track the DNA as it moves through the gel. Density makes the sample denser than running buffer, sinks into the well during loading. Color helps to see and differentiate the sample wells from the gel. And tracking the dye fronts helps in it moves through the gel at a similar rate to different sizes of DNA and can be used to estimate how far our sample of interest has migrated. So this is something, these are the different dye fronts. I was talking about bromophenol blue. Now if we talk about the type of gel, um, if the percentage of, if we talk about 1% gel, that means in 100 ml of gel there's 1 gram of agarose. Now, if we increase the percentage of the gel to suppose 2%, that means there's two, like, more amount of agarose in the same volume of solution, right? So that means it will be more concentrated, and hence the pore sizes are smaller as compared to those in 1% agarose. Okay, so for lesser percentage of agarose, lesser concentration of agarose, the, like acrylamide gels, polyacrylamide gels, similarly in case of agarose, the ones having a higher percentage will have smaller pore size, and which will be easier for resolving small sized uh, nucleic acids as compared to bigger complexes or bigger nucleoprotein complexes or bigger sized nucleic acids. We need agarose gels of lesser concentration. So the dye composition, the loading dyes composition can include tracking dyes like bromophenol blue, orange, xylene, cyanol. Some can also contain EDTA, which inhibits nuclease activity. Glycerol is added to help the DNA form a layer at the bottom of the well. Concentration of the dye can vary and may be adjusted according to experimental needs. Now, the running buffer in case of agarose gel electrophoresis can be TAE or TBE. All these stands for tris, um, which is the buffering agent. It can be either borate or acetate in case of TAE, and then EDTA for chelating ions so as to prevent nucleases from acting on the sample. Functions of TBE and TAE include ensuring the electric current flows through the gel as well as allowing nucleic acids to move through the agarose matrix. In addition, the TBE or TAE buffer maintains the pH and ion concentration during electrophoresis. The pH stays within the appropriate range because the buffer contains weak acid. And maintaining the pH is important because changes in pH can affect the net charge of the nucleic acids, and hence if the pH is within the desired range, the net charge remains the same and nucleic acids move properly. Both TBE and TAE contains EDTA. EDTA prevents nucleases from degrading the nucleic acids, and nucleases might accidentally sometimes contaminate your nucleic acid samples, and hence EDTA is the savior in those conditions. And when should you use TBE and not TAE? TBE is ideal for the following situations: when you really want to resolve smaller fragments, smaller than 2 kb. TBE actually has been found to give sharper bands, and it has a higher buffering capacity than TAE, so it works better for a longer run. When should you use TAE? It produces a better separation of larger fragments, and it works better for cloning because TBE contains borate, and borate in TBE is an inhibitor for many enzymes like ligase, which is actually an important component during cloning because we have to attach the two free ends of the DNA and vector and insert, which may be used in the later steps of cloning. TAE works better for performing DNA extraction from agarose gel. Now, DNA and RNA ladders are prepared using a variety of methods, including digestion of plasmid DNA. Plasmid with a DNA fragment is partially digested with restriction endonucleases. The plasmid should have at least 5 restriction endonuclease sites and produce at least 5 DNA fragments of different sizes. Polymerase chain reaction basically amplify those fragments. DNA fragments can be prepared using PCR amplification of desired DNA bands, followed by mixing the purified PCR products at specific concentrations. And then a combination of PCR and restriction digestion actually gives rise to DNA and RNA ladders. And while choosing the DNA or RNA ladder, we should consider the following: how many number of bands is it giving? Ladders with more bands can make it easier to estimate the size of fragments in a test sample, but the gel needs to run longer to differentiate the bands clearly, or rather resolve the bands clearly. And ladders with fewer bands may be better if time is limited and only a rough estimate of band sizes is needed. Buffers containing tris acetate EDT are commonly used for ladders with larger fragments, while those containing tris borate EDT are typical for ladders with smaller fragments. This is how the ladder looks like. Ideal DNA ladder should have several characteristics which the electrophoresis experiments need. Like every fragment of the ladder must be separable from each other. This is what is being talked about. Every fragment has enough concentration to be visualized on the gel. Here, composition of the DNA gel loading time must not affect the specificity of the DNA ladder, and definitely the ladder needs to be stable for longer period of time so that one can use it continuously. The ladder must be highly purified without any contamination. This is the same things. There can be diffusion of samples, there can be vertical streaking, there can be double bands or distorted protein bands, or there can be lateral spreading. And basically, this is what happens in Western blot. Sample is prepared, you do gel electrophoresis, then you transfer your sample onto a suitable membrane, and then you detect it using antibodies which are specific to your protein of interest, which were transferred on the membrane. Okay, so transfer methods. Proteins separated. Transfer methods can be either wet or semi-dry. So for wet electroblotting, so the most common method of blotting is through the electrophoretic transfer of proteins from the gel onto the membrane. And For wet electroblotting, the gel and membrane are sandwiched together along with the absorbent filter paper and sponges and clamped into a cassette to prevent formation of air bubbles between the gel and the membrane. And this particular sandwich is now submerged vertically in a conducting transfer buffer and exposed to an electric field. So what happens in there? The negatively charged proteins migrate out of the gel towards the positively charged electrode and become immobilized on the membrane. It is actually a high efficiency transfer method, well suited for transferring low to high molecular weight proteins, but it actually is a little time consuming. And there can be semi-dry electroblotting, which is comparatively faster. The gel and the membrane are again sandwiched together with absorbent paper in a horizontal apparatus that is placed in direct contact with the electrodes. This is mostly suited for low molecular weight proteins. So this is what is happening here. This is the transfer cassette. This is the anode, uh, like the part of the transfer cassette. This is the anode, this is the cathode. Now this is the sandwich that and that is being talked about. You have foam pads on both sides, you have filter paper on both sides. Now we have to be very careful in placing the gel and the transfer membrane. The gel has to be, the electrons are flowing from this side to this side, right? So your gel should be present towards the cathode, and the membrane onto which the samples will be deposited or transferred has to be on the positive side. Okay. And the type of transfer membrane most commonly used are nitrocellulose and PVDF, which is polyvinylidene difluoride. Nitrocellulose can immobilize proteins and glycoproteins with high efficiency through hydrophobic interactions, but it cannot be stripped and reprobed many times due to nitrocellulose fragility. However, PVDF has a high protein binding capacity like nitrocellulose only, but it is highly hydrophobic and must be pre-wet with methanol prior to use. But it is less brittle than nitrocellulose, so it can be reprobed many times. So both these membranes are available in a range of pore sizes. While 0.45 micrometer pore size membranes are generally acceptable in most experimental conditions, zero point 0.1 to zero point 0.2 micrometers pore size membranes are more suitable for blotting low molecular weight proteins. Then there's something called blocking, where we wash the transfer membrane with water to remove any non-specifically bound contaminants. And then there's probing. One can do two-step probing where two antibodies are used. Okay, first the antibody against your target protein is used, and then there's a secondary antibody which is specific against the primary antibody. And there can be either direct probing or one-step probing in which the primary antibody contains enzymes or fluorescent label for detection. This is actually not very common. Then one can even develop the blot using multiple ways, which can be chromogenic, which can be enzyme based, okay, which can be luminescent as well. Okay, so in colorimetric detection, the membrane is washed to remove the detergent, and then it's placed into a metric reagent, and protein bands appear in 10 to 30 minutes. There can be chemiluminescent detection also, and there can be fluorescent detection as well. Now let us look at some of the questions. So electrophoresis of a molecule is carried out in a solution in which type of electrophoresis? The answer is option B, moving boundary electrophoresis. Okay, in agarose gel, in polyacrylamide gel, all this are a solid matrix, right? But it is being talked about where electrophoresis is carried out in a solution. The answer is moving boundary electrophoresis. So remember where there were two electrodes, and in between there was a buffer where the sample was dissolved. Now this is a question from SDS page where what is the initiator for acrylamide polymerization? APS, which provides the free radicals. The progress of a protein sample in a polyacrylamide gel can be monitored by bromophenol blue, which is a tracking dye. Okay. Which of the following components imparts a negative charge to a protein? We read about it multiple times, which is SDS, which is an anionic detergent. In page, a layer of organic solvent is introduced. Organic solvent like isopropanol is introduced on top of the resolving gel to, to actually increase the production of free radicals, prevent the entry of oxygen. Okay. Um, so we actually don't want to increase the production of free radicals. We don't want to decrease the amount of free radicals. Okay. So we actually want to prevent the entry of oxygen so that the production of free radicals is not decreased. So the answer is option D. The pore size in a polyacrylamide gel can be controlled by manipulating the ratio of acrylamide and bisacrylamide. Okay. Which of the following component in the loading dye increases the density of the protein sample and facilitates its loading into the wells? This is glycerol. pH of the stacking gel is 6.8, resulting in the protein being sandwiched between tris and glycine, right? Because glycine amino acid will have a more or less neutral charge in this case. Which of the following can be used to monitor the folding states of a protein? Gradient urea page. Option C. So again, if we look at how gradient urea page is done, so in a typical unfolding experiment, protein is exposed to different concentrations of the denaturing agent urea, and then the structural changes in the protein can be monitored by spectroscopic or gel filtration techniques. Unfolding actually causes an increase in the hydrodynamic volume of the protein and results in slower mobility in polyacrylamide gels. In the urea page, a polyacrylamide gel is prepared with a horizontal gradient of urea. The same protein sample is loaded in different lanes and it is allowed to run vertically perpendicular to the urea gradient. Okay. As the sample runs in different lanes, it gets exposed to different concentrations of urea, and consequently at a particular urea concentration, the protein is completely unfolded with an increase in hydrodynamic volume. Right? So mostly the concentration increases from the first well to the last well. Last well has the highest concentration of urea, and hence the protein will be maximally unfolded in this particular zone. Unfolded protein will migrate slower due to increase in friction forces, and it gives a unique protein band pattern to provide qualitative or semi-quantitative information about protein folding intermediates, and the information from the gradient urea page needs further verification from other analytical techniques. In addition, urea page can also be used to analyze the protein complexes as well as covalent heterogeneity of the protein. Now, when we talk about vertical streaking of protein bands, it appears due to, why does it happen? So here, if we talk about vertical streaking of protein bands, and slide go. Hmm. So if we talk about vertical streaking, It appears due to overloading of the protein sample. It can be corrected by either reducing the amount of protein or running the gel at lower current. So vertical streaking is due to overloading of the protein sample. The molecular weight of a protein in native and STS page is 240 and 60 kDa. So if we know that protein is not a heteromer, it is a homomer, it oligomerizes and forms the complete complex, the oligomeric status of the protein will be 240 by 60 tetramer. Electrophoretic mobility of a protein in urea page decreases due to increase in the hydrodynamic volume. And when we talk about Western blot experiment, the actin protein is probed with a mouse primary antibody, mouse monoclonal primary antibody. So the secondary HRP conjugated molecule or protein antibody should be anti-mouse. Okay, it should be anti-mouse, but it should be raised in some other organism. So anti-mouse, yeah, it should be anti-mouse antibody. Okay, raised in some other organism. So the option will be horse anti-mouse HRP. So that's it for today's lecture. Thank you for joining.